Hi, everyone. Welcome to Lunch with a Scientist. This is Leah with Headwaters Science Institute, and today I'm very excited to welcome Inga Conti-Jerpe. She's a coral reef ecologist with a primary research interest in how corals obtain nutrients and food and survive and reproduce. Inga has a PhD from the University of Hong Kong in Hong Kong, China, where she used stable isotope analysis as a tool to investigate variation in coral nutrition across species. She's passionate about science education and outreach. She loves helping others investigate and understand the natural world. She is an avid scuba diver outside her science research, and she got her certification when she was 14 and has logged hundreds of dives for research and for other purposes. So she's very passionate about coral reef research, and she's going to be here today to tell us a little bit more. So I'm very excited to welcome Inga. Hi, how are you?
I'm great. How are you?
I'm great. And where are you coming to us from today?
I'm actually in San Francisco, where I recently moved only a couple months ago from Hong Kong. Yeah, big change.
Fantastic. Interesting. What's it like having come back from a different country where you were doing all this research?
The biggest pro is the weather. Hong Kong is incredibly humid, and I'm very happy to leave that behind. But I do miss I miss my lab and the people I worked with there. They were really the main attraction, working with some great people. Yeah.
Well, we're super excited to hear about some of that research, so I'll let you pull in your talk and I'll let you take it away.
All right. Thank you so much. I'm so excited to be here today and have this opportunity to talk a little bit about some of the research that I did during my PhD. And really, this is just one branch of the research that I did, but I was really excited to be able to look at coral nutrition and quantify it and look at its relationship with bleaching resistance. But before we get into the science, I wanted to talk a little bit about my background and the path that led me to where I am today. So I started off my education with getting a bachelor's of science at Cornell University, and I've always loved the ocean. I got certified to scuba very young. I learned how to snorkel when I was 5, but during my undergrad, I was excited to explore and try different things. And so I turned towards environmental science and some of the policy aspects involved with that. So here's a picture of me working in a small plane to do surveys for a job I had with the National Park Service. I also was able to travel to other places like Kenya and learn about the ecology there. And I did my undergraduate thesis on freshwater food webs. So it was a little bit of a divergence for me, kind of exploring some other ecosystems, but it gave me a really strong basis for what I did moving forward. So then I went on to the University of North Carolina, Wilmington, where I did my master's. And I was really, really fortunate because the lab I was in there used the underwater habitat Aquarius reef base for their research. So I got to spend 10 days there working on a project tracking populations of sponges on the reef. For my own work, I decided to focus on sea fans that you can see in this image down here that are kind of understudied corals. And I was interested in what they were eating. And so I took these animals into the lab and tried to feed them different food sources, but it was really difficult. And so that's when I first learned about stable isotope analysis, which I'll talk a little more about later on in the talk. So then I had the opportunity to go on and do my PhD, and I was really excited because I found a place that combined lab work with field work, and I found an advisor who was really supportive in letting me pursue the questions that I wanted to ask in science and develop my own research questions and move my own research forward. The big change was that this was in Hong Kong. And so here I have a map where I've put a little pin where Headwaters is located in Northern California. Hong Kong is on the other side of the Pacific on the southern coast of China on the South China Sea. And this is a city of about 7.5 million people in a relatively small area. So there's a lot of impact from humans on the environment. But despite that, they have these beautiful, vibrant coral reef systems. And I was super excited to be able to study them because they have more coral species in this one small area of Hong Kong than the entire Caribbean Sea. So this was my opportunity to go to a new place and learn about a new system and learn about these new players in these systems. So why do people study coral reefs? When we look at a picture of a coral reef like this, the obvious dominating factor is this very diverse and textured sea floor, which is built by the corals themselves. And we can see that there's lots of different structures that the corals are building. Some are smooth, some are branched, and this provides really diverse habitats for other organisms to live in. Here we have a map of where corals occur around the world. We have both warm and cold water coral reefs. And warm water reefs tend to be a little bit more diverse. So if we took all of these red points and added up the area where coral reefs occur, we would find that they exist on between 1 and 5% of the ocean floor. But because of this special habitat that the diverse coral structures provide, these reefs support 25% of global marine biodiversity. So that means that 1/4 of all the species that occur in the oceans occur in coral reefs. So coral reefs are these incredible places to study the interactions between species because we have so many species coexisting in such a relatively small area. So some of the questions that ecologists ask generally are, how can so many species coexist? And this is obviously particularly relevant to coral reefs. What is special about coral reefs that allows such high diversity compared to other ecosystems? We also ask questions about the interactions that species have and how those shape communities. And we're also interested in what governs species interactions. What are the evolutionary pressures that result in these interactions? What are the adaptations? So I'm going to focus on the first question, how can so many species coexist? And to understand this, scientists have developed the concept of the ecological niche. And this is the conditions and resources that a species requires to survive and reproduce. So here we have an example of a barnacle, which is an animal that grows attached to rocks in the intertidal zone. And we can see that this one species of barnacle, Thalamus, lives all throughout the intertidal zone, from the low tide to the high tide. The higher you get in the intertidal zone, the more you're exposed to air, and the more stressful it can be for these animals that find drying out really stressful. But this species has a very broad ecological niche, and it can tolerate these very dry conditions. If we add another species into this ecosystem, Semibalanus, we find that Semibalanus is really dominating the lower intertidal zone. And so these two species are competing with each other, and Semibalanus is winning. However, when we reach the high intertidal, Semibalanus can't tolerate those dry conditions. And so Thalamus is able to survive and carve out this space for it in this ecosystem. And so this is what we would call niche partitioning. These two animals are able to coexist because they have different ecological niches. And so this is one of the ways that scientists think that species can co-occur. There is a subset of the ecological niche called the trophic niche. And these are the food and nutrient resources that a species needs to survive and reproduce. And a really great example of how the trophic niche can be partitioned are Darwin's finches. So these are finches that live in the Galapagos that all have evolved to use one very specific trophic niche and one specific food source. And that's reflected in the structure of their beaks. So here we have all these finches with different beak structures. And this guy down at the bottom has a really large beak. So perhaps he has evolved to crack open really tough, big seeds. But then we have other finches, like this little warbler who has a small beak. Maybe that's really great for picking up tiny ants. And so this is a good example of how these different finches have adapted to use different trophic niches and avoid competition with each other. So does that occur in corals? How do corals get the nutrients that they need? So here we have a coral colony, and if we zoom in, we can see that this colony is actually made up of tens, hundreds, or even thousands of individual coral polyps. And each polyp is an animal that can exist on its own, but they live and work together in these colonies. So here we have a close-up of the polyp, and they're animals, just like humans. They have a mouth, they have a stomach, but they have some other more unusual aspects. They also have tentacles that surround the mouth that they use to capture particles and eat. And then they also have a skeleton, an inorganic skeleton, and that kind of forms the underlying basis of the colony. And that's also what provides that structure in coral reefs that the corals build themselves. Finally, the polyps are all connected by this living tissue. And so that means that can pass nutrients to its, and so this these individuals can share throughout the colony and really kind of act as a unit overall, despite the fact that there's so many individual animals living within it. So corals are animals, these polyps are animals, and so they can eat. And so what corals eat are plankton, small algae and animals suspended in the water column. They capture them with their tentacles and pass them into their mouths and digest them in their stomachs. And this is a form of predation. And we would consider this to be a heterotrophic food source. And so heterotrophic food sources are where you can't make your own food. You have to consume other plants or other animals to get your nutrition. So humans are a great example of heterotrophs. Corals, on the other hand, have associated algae in the family Symbiodiniaceae. And so these are tiny microscopic unicellular algae that live inside the coral tissue. So here's a close-up of another coral polyp, and we can see this kind of green, brown, yellowish color. Those are the symbiotic algae inside the coral tissue. And if we take an even closer look, what we see is that the symbiotic algae, these little brown splotches, are living inside the cells of the coral host. So this is an intercellular relationship, and they're very closely intertwined. So algae, like plants, can harness light and produce and perform photosynthesis, and they can pass the products of photosynthase, that is typically carbon or other organic nutrients. They can pass those to their coral hosts. In return, corals provide their algae with waste nutrients. And so just like you can use manure to fertilize plants or crops in a field, these algae can use the waste from corals, and then we convert that back into an organic form that the coral can use again. And so they can actually recycle nutrients between these two partners. And this is a symbiosis. So both the algae and the coral are benefiting from this relationship that they've evolved to maintain. So because algae can make their own food through photosynthesis, we would consider that an autotrophic food source. They don't really need to eat to meet their energetic requirements. And what coral scientists started to realize is that the trophic strategy of corals can range from almost completely autotrophic to completely heterotrophic along a continuous spectrum. So some species of corals seem to be highly dependent on their symbiotic algae. They really need them to survive. But other corals lack this algae completely, and they only feed on plankton. And this spectrum ranges through all different combinations of these two trophic sources through different forms of mixotrophy, meaning just a mixture of the two. So we now know that there are these two different nutrient sources that corals can access, and that they have different trophic niches that kind of range between these two extremes, autotrophy and heterotrophy. But is there a structure that's kind of the coral equivalent of a finch beak? Is there some kind of part of their anatomy that is indicative of what trophic strategy they use? And so one idea that's been proposed in the literature is that polyp size is a key adaptation to autotrophy or heterotrophy. And the idea is that a small polyped coral has a high surface area to volume ratio. And so this provides more surface area to intercept light and perform photosynthesis. On the other hand, large polyps have less surface area for their volume, but they also have a larger polyp. At the center, there's a larger mouth, and also inside there's a larger stomach. And so this kind of implies that a larger polyp is able to consume larger plankton, and it increases the range of plankton a coral can feed on and makes it a more viable and more accessible resource for large polyped corals. So I wanted to answer the question, in my research, I wanted to answer the question, is polyp size an adaptation for different trophic strategies? But it's quite difficult to estimate the trophic strategy of a coral. We know that it varies, and certain species have been studied, but it's difficult to assess trophic strategy over a broad diversity of species. And that's because it's really difficult to measure how much plankton a coral eats. It's hard to count how many microscopic particles they filter out of the water. And it's almost impossible to measure how many nutrients are passed from those algae inside the coral cells themselves. So to answer this question, I needed to use a different method to measure how much corals relied on these two nutritional sources. And the technique that I used is called stable isotope analysis. So here we have the box of carbon that we've removed from the periodic table. And carbon has this little 6 in the upper left-hand corner, indicating that carbon is defined by having 6 protons in its nucleus. And most carbon atoms have 6 neutrons, which means that their mass is the sum of those two numbers for a mass of 12. However, an extremely small subset of carbon atoms have an extra neutron. They're still carbon because they have 6 protons, but they're slightly heavier. They have a mass of 13. And so carbon 13 is a stable isotope of carbon. Nitrogen has a similar situation. Nitrogen is defined by having 7 protons in its nucleus, and it typically has 7 neutrons for a mass of 14. But a small subset of nitrogen atoms have 1 extra neutron and a mass of 15. So we can use the relative abundance of these heavy isotopes to map and understand food webs. Now, to understand how we can do that, we have this biplot here where we can plot our isotope values. And on the x-axis, we have our carbon isotope value, and on the y-axis, we have our nitrogen isotope value. Now, here we have this delta symbol, which is the typical notation that scientists use when they're discussing isotope values. And a very useful phrase to keep in mind when interpreting these values is higher, heavier, lower, lighter. So as we have a higher isotope value, we have more heavy isotope, and as we have a lower isotope value, we have less heavy isotope. So on this biplot, we can superimpose a simple food web. So here we have phytoplankton, which is kind of like algae plankton, like kind of a plant version of plankton. And those get consumed by zooplankton, which are tiny microscopic animal plankton. And those get consumed by a heterotrophic coral. And we can see that as we move from one trophic step to the next, as one organism consumes another, the carbon values are not changing. We stay in this light range up each trophic step. But we can see that nitrogen is increasing a lot. And if we look across ecosystems and all different species, it turns out that these increases are predictable. So across ecosystem, across species, carbon either doesn't increase or increases at most 1 per mil. And nitrogen increases between 2.5 and 3.5 per mil. So we have this expectation for what our heterotrophic coral will look like based on this little food web. And we can compare that to what we might expect for an autotrophic coral. Because these corals are getting their nutrients straight from their symbiotic algae, we would expect their nitrogen values to be much lower. And we would expect a mixotroph to be somewhere in between since it's using both of these sources. So what we can do is we can go out into the field and we can take little samples of a whole bunch of individuals, of colonies of a coral species. We can measure their carbon and nitrogen isotope values and plot them on a biplot. And we can fit this ellipse to them. And this ellipse is what we would consider the isotopic niche. And because of how isotope values change through food webs, it's a proxy for trophic niche. And so here we've measured the isotopic niche of the coral, and we can compare that to the isotopic niche of its associated algae. Now, in a heterotroph like we have here, our hypothesis is that these isotopic niches would not overlap, indicating that the coral is accessing different nutrients than its algae. But in an autotroph, we would expect to see a high amount of overlap between those two groups because they're sharing the same nutrients or cycling them passing them from one to the other. So we sampled 7 species of corals from across Hong Kong. And we tried to target here, this is a map of Hong Kong. And each little triangle is a site where we collected up to 3 individuals of each species, if it was present. And we tried to target a range of species that showed a diversity of polyp sizes to try to answer this question of whether polyp size is related to trophic strategy. So I'm going to present the isotope data to you. Again, we're going to have our nitrogen value on the y-axis and our carbon isotope value on the x-axis. And what we found is that some species have an extremely high amount of overlap between the host and the symbiont isotopic niches. And we classified these as autotrophic. They're primarily relying on those photosynthetic sources of nutrition for their survival. On the other hand, we had several species that showed little to no overlap between coral host and associated symbiotic algae. And these we classified as heterotrophic, suggesting that they feed more on plankton. And then finally, we had two species that were kind of in the middle. They had partial overlap of these two isotopic niches, and we classified those as mixotrophic. Now, another way of thinking about the difference between host and symbiont isotopic niche is to look at the distance between the centers of those two ellipses. So for Acropora, our most autotrophic species, we can see that the centers of the ellipse look really, really close together here. And if we measure it, it's about 0.3 per mil. Platygyra, on the other hand, the centers of these ellipses are quite far, and it turns out that it's about 2.5 per mil. And so what we did is we plotted the distance between the centers of the host and symbiont ellipses on the x-axis down here. And we looked to see if there was a relationship between that and polyp area, which is a proxy for the size of the polyp. And what we found is that our autotrophs had relatively small polyps, which we would expect. That supports our hypothesis. The mixotrophs also had pretty small polyps, but our heterotrophs had much larger polyps. And this relationship was statistically significant. So we ran a regression analysis that demonstrated that these two are significantly correlated with each other. So this provides the first really robust piece of evidence that this hypothesis about polyp size is true, and that large polyps are adapted for heterotrophy and small polyps are adapted for autotrophy. So we think that this may be part of the reason why corals are able to coexist on the same reef. They have this trophic niche partitioning. Some are using autotrophy, some are using mixotrophy, and some are using a combination of the two. But we wanted to take this a step further and think about how the trophic strategy of coral species affects how they interact with their environment, rather than each other. And we decided to look at coral bleaching. And so, as many of you might be aware, bleaching is a phenomenon that's occurring more and more as a result of global climate change. And this is when sea surface temperatures elevate just as little as 1 degree above the mean temperature in the hottest month of the year. And corals, in response, turn a bright white color. And so what's actually happening when these corals bleach is that they're expelling their associated symbiotic algae. So the algae are what give the coral those brilliant, bright colors. And when the temperature starts to increase, this affects the photosynthesis of the algae, and they start to produce products that damage the coral. And so the coral expels them to try to protect itself. That removes the pigment and just leaves clear coral tissue through which we can see the bright white skeleton that lies underneath. And so when we have bleaching, we're essentially removing one of the pathways that corals use to obtain nutrients. They can no longer get those photosynthetic products from their associated algae. So I was curious, does trophic strategy play a role in how susceptible corals are to bleaching? It seems like bleaching affects one of these pathways. And so if a coral is more able to use heterotrophic sources and feed on plankton, perhaps it is less susceptible to bleaching. So to test this hypothesis and to answer this question, we ran a warming experiment. So I had 7 nubbins of 8 different species in this array that you can see here. And so each one of these cups holds an individual coral nubbin, and it's sitting in a water bath, where we slowly increase the temperature over time to mimic what would happen on an actual reef in the field. So here we have the temperature of the experiment shown in this orange line, and you can see that there's some variability, but in general, increased over the duration of the experiment this shows around day 20 to day 70 at the end. And then we also have this yellow line, which is degree heating weeks. And this is a measurement of how much thermal stress the corals are experiencing. And it's not just how high the temperature is, but also how long the corals have been exposed to it. And so we can see that this thermal stress is increasing fairly consistently over the course of the experiment. So we can zoom in on the timeline of the experiment from day 45 to 70 and look at when each species of coral bleached. And what we found was that our autotrophs bleached first. We had 3 autotrophic species that bleached earlier in the experiment. We had 3 heterotrophic species that bleached later. And in fact, there was even one species, Phovites abdita down here, that didn't bleach fully even by day 70. So we can't include it on this graph, on this figure. And then we have 2 mixotrophs. Now, Porites lobata fell right in between our heterotrophs and our autotrophs, like we would expect, But we had this other species, Pavona decusata, that was bleached right at the end of the experiment. And we were pretty surprised by that. We had this really nice pattern in all of our other species where trophic related. So what was Pavona? It was trend. So we took a little dive into the literature to try to understand what was happening with this oddball. And we realized that Pavona was the only species in our experiment that had exhibited a vertical plating structure. So it kind of grows in these plates oriented towards the surface of the water. So here we have a picture of a field of Pavona in Hong Kong. And you can see these plates that kind of grow upwards. And bleaching is controlled not only by temperature, but also exposure to light. So you can imagine that a coral kind of growing with this vertical plating structure has more shading on either side of it than a coral that's shaped like a dinner plate facing the surface of the water. And so we wondered if this type of growth form was conferring protection against bleaching for the species. And so we did a little digging and we found a paper in the literature that looked at a different species of Pavona that had bleached in the field. And this has a slightly different structure. So it looks more like a table with a flat top and then vertical sides. This picture's pretty grainy. It's from an older paper, but you can see that the tops of the corals are bright white. They bleached completely, but the sides of the corals still have a lot of pigment. So those vertical sides were protected and were able to withstand bleaching a lot longer than the top surface. So we took this as pretty good evidence that the structure of this colony was affecting its susceptibility to bleaching, in combination with its trophic strategy. So we kept that in mind when we did our statistical analysis. So here we're going to show the relationship between the distance between the ellipse centers of the host and symbiont from our isotope data for each species, and then the time it took for a species to bleach on the y-axis. And what we found was that there was a pretty compelling trend with, as we expect, Pavona being this outlier. And we ran our regression analysis with all species included, and it was not significant. But when we excluded Pavona, it was. And so we think that this is strong evidence that trophic strategy is related to the bleaching susceptibility of a coral, but there are obviously other factors that influence how easily a coral bleaches, including its structure and what it looks like. So there's more to consider when we think about this story of what makes a coral more or less likely to bleach. It's not just trophic strategy. There are other factors as well. So here we have our schematic showing these two different extremes of trophic strategies in corals, and the polyp size being one of the key characters related to it. And we can add bleaching susceptibility to this diagram. And so this is a rough tool that people interested in conserving reefs can use when they're trying to make decisions. So we can associate small polyp corals with being bleaching susceptible. So it's kind of a quick and easy way to get a rough estimate of how likely a coral is to bleach and how susceptible it is to climate change. So when we go to a reef that looks like this, that's dominated by these branching corals that have very small polyps, this is a reef that's going to get hit really hard by a bleaching event. These are autotrophs that are likely going to bleach early and be less likely to recover from bleaching. And so when we're deciding how to best protect reefs moving into the future, maybe we want to set aside and protect reefs that are dominated more by heterotrophic corals and are more likely to survive as climate change continues. We also want to think about this when we're restoring reefs. So around the world, there are different organizations that are growing corals in nurseries and then outplanting them back onto the reefs. And they have predominantly emphasized branching species with small polyps, mostly because those are the species that have started to disappear most quickly as climate change continues. But as we invest time and resources into these projects, we might want to consider targeting species with larger polyps that will be more robust as we continue to combat climate change. So these are some of the implications of the work that I've done that hopefully can help us better protect reefs and restore reefs moving forward into the future. So with that, I'd really like to thank my funding sources. Several grants from the University of Hong Kong and the Hong Kong government supported this work. And I had many collaborators and team members at HKU and the SWIMS, the marine lab there, and my lab that helped this work. It would be impossible for it to be completed without lots and lots of help. And thank you so much for taking the time to watch this talk. I hope that it um, help you understand how trophic niches work and the trophic niches of corals, and how trophic niches help us understand how species interact with each other and with the environment. So thank you so much. I'm so happy for this opportunity.
That's great. Thank you so much, Inga. So I have a few questions that I want to jump into. First off, I know that you've done a lot of work out in the field, which for you is underwater. But what does being a coral scientist in its entirety look like when you're not underwater? Are you harvesting pieces to bring back to the lab? And how do you look at those pieces and study them?
That's a great question. So there are experiments that happen in the field, but they are the most challenging, as you can
Mhm.
imagine. Getting things to work underwater is always a challenge. And so a lot of our work is, collecting specimens for experiments and bringing them back. We use a lot of tools that biologists in all different fields use to understand corals. So we use molecular tools using DNA sequencing to look at populations of corals, how they're connected. Some people are starting to look at gene expression in corals, and if uh, certain species have genes that can help them deal with certain changes in the environment. We also use, do experiments with live corals in labs. That's a lot of maintenance, a lot of tank cleaning, a lot of feeding your corals and making sure that the water chemistry is perfectly correct. And of course, with all science, a huge part of it is reading and writing. You spend a lot of time reviewing the literature, staying updated on the literature, and interpreting what's there through the filter of your own work. How do these other findings relate to what you did? Did they confirm or refute your findings? And how can you make sense of that? So I think that the field, I think, is what a lot of marine ecologists love, but it's a small subset of what we actually do.
Mhm. I see. And so I wondered, when you're looking at these coral polyps, does every species have the same polyp size, or at least same expected size, or are there ever any outliers?
You mean individuals within a species?
Mhm.
That's a great question. So there is a lot of what we would call morphological plasticity in corals. So this just means that under different conditions, they can look very different. They have
Mhm.
this arsenal in their DNA where they're like, oh, the current changed. Now I want to grow like this so I can better catch food. Or, oh, it's uh, maybe there's more light. I instead of growing up to reach the light, I should grow out. And
Mm.
so they really do have a lot of morphology. I think polyp
Mhm.
size is something that we're learning more about. There's not a lot of data out there. And in general, the community right now treats polyp size to kind of be like a standard thing for a species, like a standard trait. But I think one of the strengths of the work that we did is we measured the size of the polyps of the corals we had isotope data for. So that was really important. I spent a lot of time digging up polyp sizes of like bleaching data from like other places, trying to get these correlations. But I think what you're saying is exactly true, where their polyp size is not always the same
Mm.
because they may be using heterotrophy or autotrophy more within a species. And I think some species are likely to be able to switch and others maybe can't. So corals are really complex,
Mhm.
and they have a lot of factors that we have to consider. And I think that's part of the reason why we're still learning some of these things about them that seem really basic. Like, what does it eat? That should be easy, right? But like, no, it's actually really challenging.
Mhm. So I'm also wondering, we're talking about some expected research and the standards, and then some new research here. How much of this research that you're doing is brand new, and how much of it is based on previous research that scientists have done before?
But I mean, to a certain extent, it's all based on stuff people have done before, right?
Mm.
So there, it's we the marine ecology as a field is is on the shoulders of terrestrial ecology.
Sure.
We were a much newer discipline because we really couldn't study the oceans until we had SCUBA in the 60s or 70s. And so terrestrial ecology is much older. And so we
Mhm.
steal a lot of ideas from them and test them
Mhm.
in the marine environment. But yeah, so the idea that polyp size, for example, is related to trophic strategy is something that's been in the coral literature, I think, since the 70s. It's just that nobody has it's debated. There's a little bit of evidence here, a little bit of evidence here. This is really the first paper that shows across a broad range of species that, yeah, this looks like this is a
Mhm.
real relationship. And so a lot of science is taking ideas that have been tossed around for decades and, applying them in a new way. Isotope analysis is a really old technology. But
Mhm.
again, we're learning new ways to use it. The fitting that ellipse is new, something that I didn't develop, but I applied it in a new way, right? Most people don't compare symbiotic partners with that. So that's a new use of that analysis. So the newness of this work is kind of small when you consider all of the existing knowledge necessary to make it possible. Yeah.
Yeah, I think that it's fascinating. It's a coming together of lots and lots of generations of research, which is really, really interesting. And so, as we move into kind of your analysis portion of the research, where we can talk about coral bleaching and what to do about it, I know that it's become a hot topic in today's world for sure. Can you dig a little bit deeper into any kind of recommendations that scientists may have for how we can prevent these small polyp corals from damage, knowing that they're more susceptible?
Yeah, there's a couple of key points here. So the first is that humans need to address climate change.
Mm.
And I would say that the number one thing you can do is write your politicians and make sure that they know that it's an issue that you care about and that you want to see action on. People can take a lot of steps independently to reduce their own carbon footprint, and I think that that's great and worthwhile. I take a lot of steps. So, for example, this is really out of a lot of people don't associate these things, but try to eat less red meat because beef has a really high carbon footprint and contributes to greenhouse gas emissions. So you can take lots of little steps in your own life, but I think the biggest thing you can do is try to motivate systematic change at the national level. um, there's also a lot of work out there that shows that other stressors from humans and other impacts can kind of synergistically work with global climate change and make the situation worse for a local reef. So there's a couple of really big papers that show that nutrient pollution and runoff from construction, these kinds of things, if you have those and then you have a bleaching event, it's way worse than if you didn't have those stressors. So local stressors still really matter. And so again, this is something where if you live near the sea and you go on vacation, maybe try to support places that are more conscientious of their impact on the surrounding reefs. If you visit a reef, don't stomp on them. That's a big one that people say. But yeah, so a lot of those local stressors matter too. So I think a lot of times the future of corals can feel overbearing because it's this big global problem that we're facing with climate change. But we shouldn't let go of the changes we can make locally because they also really matter. One that I think a lot of people don't consider is supporting sustainable fisheries because overfishing in reefs can make the whole ecosystem go out of balance and add extra pressure. So a lot of those things, kind of when you have the opportunity to interact with a reef or to be near a reef, maybe keep some of those things in mind and try to support practices that protect the reef as much as possible at the local level.
Mhm. Thank you so much for sharing. There you have it, some recommendations from scientists about how to protect our oceans and our coral, which are so, so important to our ecosystems. And so, Inga, I want to wrap up just with some inspiration for our students.
Mm,
If there's a student out there that is interested in the ocean or maybe fascinated by coral, but doesn't quite know what they want to do with that as a career, what
mm.
would you recommend they take as an educational path?
So, I mean, I think you can take a lot of tools from science in general and bring them to a marine system. I think a lot of people kind of pursue science because they're passionate about one specific system, but the skills are transferable. And so, you know, I would say any opportunity to make observations about nature is like a great step number one. In your own backyard. I've been in the house for COVID. I've started bird watching out my backyard, and I'm learning about different hummingbird species and how to tell them apart, what what their are. So learn about always marine environment. But if you go to school, if you go to university, you can always try to go to a school that has a strong marine program. But even if they don't, freshwater is everywhere, and they're very similar systems and have a lot of the same kind of questions that we ask. Same with terrestrial systems. So I think getting your hands dirty, getting outside, and learning about how ecosystems work overall is a great way to get into ecology. And then later down the road, you can always target marine if that's what you're really interested in. Yeah.
Great. Well, thank you so much for presenting. We're so thankful for the work that scientists like you do and thankful for sharing a piece of that with our students today. So,
Thank you so much
Inga,
for the opportunity. I love trying to uh, disseminate science as much as I can. It's a really great opportunity.
great. Thanks so much.